The Metabolomist is the podcast where scientists connect to explore the unspoken issues in metabolomics research. I am your host, Alice Limonciel, and together we will examine how metabolomic data interpretation is done. I will discuss with other metabolomists how they do this work, how they plan, execute, but also communicate metabolomics. Welcome to this podcast. Thank you for discussing with me today. I will start by introducing you with a short biography, and then we can discuss your work with metabolomics in further detail. But it was really interesting to look through what you did over the years. You studied mathematics and physics at the University of Osnabrück in Germany. And then suddenly you had a PhD in atmospheric chemistry and meteorology.
Yes, but that wasn't suddenly. I mean, during my studies, I was half a year in England and studied fluid dynamics there. And then fluid dynamics led to meteorology.
Okay.
And then the specialization in meteorology was atmospheric chemistry. And in the end, honestly, it's not very different from what I do today. Atmospheric chemistry is like the metabolome of just one organism, which is the globe.
That makes sense. So it's all about the analytical methods. I get it now.
Well, I could pretend that in reality, it's a lot of personal reasons.
So you did your PhD in Toulouse in France, and then a few years later, you habilitated in bioinformatics and structural biology in 2004 at the University of Aix-Marseille, and you stayed in France for a few more years until 2006, where you were appointed professor for bioinformatics at the University of Munich and Helmholtz Center in Munich in Germany.
Yeah.
And in 2011, you joined the Department of Physiology and Biophysics at Cornell as a full professor and became the director of the Bioinformatics core at the Cornell campus in Qatar.
That's right.
As I was reading through your profile page on the Cornell website, there's one sentence that caught my attention. You wrote, "I identify as a bioinformatician and system biologist." And if you remember writing this, I would like to ask you first, why did you choose that verb? Why do you identify as a bioinformatician? I mean, you're a professor in bioinformatics. I found this really interesting.
It's because in a way, I never really liked to be boxed in somewhere. So when I was a physicist, I was always saying, no, I'm more a theoretical physicist, a mathematician. I'm computational stuff. I mean, I'm always evading things. And at some point, I felt maybe now I arrived somewhere. And so I said, because being a bioinformatician is also some kind of, the definition of bioinformatics also changes a lot between countries. For some people, it's just computer guys. For others, basically, and that's more the interpretation I like, is it's basically the study of information content in biology. And I think that's the definition that the NCBI gives on their webpage. And that's something I identify with.
Something that fits. You identify with this at the moment. I love this choice of word, because it's also nice to see you get the degrees, you get the job titles, but still you can still identify as whatever you like to identify. And then another thing I found interesting going through this bio is that, so it changed quite a lot.
The driving thing was just like the curiosity about how biology actually works, because being a physicist, initially, when I studied, biologists were frowned upon. And when there was just the human genome sequenced, it changed everything. Suddenly, biology was really mechanistic and much in front of it. I changed totally about this view.
So this is what pushed you towards biology then also, when the genome was...
I mean, it was a chance event. I was with CNRS. I had only a detachment, so I could go back. So I went to industry in my hometown for personal reasons. Did engineering for 2 years, and seeing engineering and research is not the same thing. The one guys know exactly what they want, optimize things, and the others, the researchers, don't. And I didn't fit in there. Luckily, I was in CNS, could go back. Theoretically, I would have gone back to atmospheric science. But I just by chance, I ran across this kind of paper in Le Monde on the discovery or the publication of the human genome. And when I was just applying for a position to go back to the region of Marseille, I said, well, maybe I just ask him. And it turns out that in his lab, all bioinformaticians were physicists, astrophysicists. He was a physicist. So at that time, basically, there were no bioinformaticians. They were all converted biophysicists. So that was really the driving thing.
That developed later then. The job of bioinformatician, that the label didn't even exist probably at the time, or it was really a small niche thing. Or
Yeah, I mean, it was really just the discovery. I mean, the problem of really solving the sequencing problem. They had all the sequencing, but having the alignment algorithms, all that was really what bioinformatics initially was.
Mm-hmm. Genomics is still a big topic now, but metabolomics is a topic that grew a bit later. It seems like for the last 15 years, you've been quite interested in metabolomics, and that this still keeps you interested. Is there a reason for this?
Yeah, it's a functional thing. I started biology or bioinformatics in 2002 when I went to Marseille and learned it from scratch in that lab. And already then I had a colleague, Hiro Ogata, who was actually the first author on the first paper of the KEGG metabolomics pathway map. And he was working in Marseille at that point. With him, I learned to analyze the metabolic capabilities of bacteria. So we sequenced different bacteria and the way of looking how intracellular bacteria live. So the kind of genes they lose, they become dependent on the host cell, that you could computationally analyze that. He was really there. You just took the million base pairs or whatever you had in a bacteria and broke that down in all the enzymes. And then he could show which ones are missing and which bacteria does use what from his host. They were all obligate bacteria. So there was already metabolomics in a certain way, although I didn't know it at the time. And the other thing is the link between the function of what the genome really does. How does it really function? How do you interpret that? And I think these were the first steps where I could interpret the function in a bacteria. And that later carried on when we went to Munich, where the real breakthrough, not the breakthrough, but when things really got into humans to say, how is human metabolism working, genome-wide association studies, and then doing the mGWAS with metabolomics. I think I was lucky to be at the right place at the right time with the right colleagues around me and everything. So
it's usually how it happens.
Yeah.
Yeah, and then this started a series of really nice papers. This is the main thing I wanted to discuss with you today. I'm not going to discuss the detailed statistics behind it. I think it's interesting for people to have a global understanding of how this is done, because genomics and metabolomics are two very different data types. So it's interesting to discuss this, but also to see what metabolomics brings in that environment. Maybe I can give you my very superficial view of it, and then you can give a bit more detail, because one of the most interesting things for me was that there seems to be people who start from the genomics and then the SNPs and then put their phenotypic trait, whether it's metabolomics or something else, on top of it and look at where the associations are. And there seems to be another technique where you start from the phenotype, whether it's metabolomics or something else, and then you find what's interesting there, and then you find what is associated with it in the genome. Did I understand that correctly, firstly? And can you comment on this?
I think you should go maybe further back about the mGWAS. I mentioned already the Human Genome Project being at the beginning of everything. Not at the beginning, but that was really when things really took off. And the Human Genome Project was already showing that there is genetic variance between people. I mean, it wasn't the genome of one person, but I think at least 10 different genomes were put together. And the promise of the Human Genome Project was, we just go and find all the variation and associate that with the outcome. And once we have that, we can treat everything.
Yes.
And of course, things turn out to be much more complex. And some people say it didn't work. I would not agree with that. Absolutely not. But some people wasn't worth it. In any case, the mGWAS came and were like initially say, we would just find the gene for diabetes and the gene for this and that. But realized it's not true. You just can explain 1, 2, 3% of what you suspect to explain of the heritability. Nowadays, we know much more about variants out there. A lot of small effect sizes with many variants and some rare variants with large effect sizes. And I think that could go on that. The one other thing that came is a question, what do we do with mGWAS? What's the purpose? And I think a misunderstanding is that people think, or initially maybe sold like that you can predict something. You can predict whether someone dies of this or gets that disease. But it's much more nowadays about understanding, on the one end, the pathways. And I think from a pharmaceutical point of view, also target validation, target interpretation. For them, it's important if they want to work on a molecule inhibiting a protein, that there's genetic evidence that if I tinker with that protein, something happens. And that's where the metabolomics comes in. I think this concept of what we call the intermediate phenotype. And I think that has been something that has been pretty early on in our first GWAS papers. I think it was Florian Kronenberg from Innsbruck who actually brought it up, is to say these intermediate phenotypes, they're the link between the genome and the disease. And it's not only metabolomics. And that's also why I think there's no priority for metabolomics or any other omics thing. The whole chain from the genetic variants, which is the starting point of the GWAS, and the end point, whether someone gets a disease. And on the meantime is all the other genetic variation that's influenced by the genetic variant. And you measure that in metabolites and proteins. There's GWAS on glycosylation, on lipidomics, on microRNAs, on anything you want, on methylation.
Yeah, and this grows as the techniques grow as well, like as the technology improves for glycosylation or things like this, then it's more applicable to associate with other omics, I guess.
Right, and that makes the value of, now if we go back to metabolomics, I mean, metabolomics is measuring the true endpoints of biological processes, which is maybe also a little bit of an exaggeration, whether they are the true endpoints, but it was like you go from the gene to the RNA to the protein, and then the metabolite is where things happen. Of course, diabetes is glucose and gout is urate. So in the end, it's true, but there's certainly other things where...
Yeah, but this is the old paradigm as well. That's more and more we see interconnections between the different levels, and then it's even difficult to see the levels anymore because everything is everything now. Is it correct that in your papers you start usually from the genome and then add on the other methods, or at least the metabolomics, or do you do both versions? Is there an advantage to one or the other?
No, it's always together. I mean, association study
just
means
together.
one is one is the dependent, one is the independent variable, but that's just artificial, which you take as dependent or independent variable. It's in the end, it's a correlation.
Yeah, but let's say if you're looking at cohorts with diabetes or without diabetes, then you might use one data set to focus on the differences between those two groups and then add on the second layer. That would change some of the results, wouldn't it?
Yeah, but that's a different kind of study. I mean, you also, when you do metabolomics, you should distinguish what you're studying. Now, we started out with GWAS and GWAS with metabolomics because that's a bit what I was focusing on. But in general, you don't do metabolomics for genetics. There are two kinds of studies. The ones are these population studies, and that's a bit where we were working on, like the Cora or the Framinghams and UK Biobank. So where you take everybody who is more or less normal, and then you collect as much phenotype information and genotype information, everything. And then you can do all against all. And on the other hand is more these kind of clinical studies. Like say, I want a cohort of diabetics and see what's the case control or kidney rejection and things like that.
See. Yeah, that makes sense.
And in terms of data acquisition, it makes also a huge difference because in the one case for the population studies, it's really like generating this data once and for all in high throughput for thousands of samples. In the other case, it's more like you spend less money because there's less samples, but you enrich them in cases and you have more detailed phenotyping, you have much more details on the patient and stuff like that. And in an ideal world, like with UK Biobank, you have both. You have so much detail on them that you can do whatever you want. And then you just filter out like the diabetics and the non-diabetics from UK Biobank and do your case control study and this kind of things.
Okay. So let's go back to what I think the first mGWAS paper was the Gieger paper from 2008. Is that right?
Yeah, Christian Gieger.
Could you tell us a bit about that paper and maybe your role also at the time? I'm interested in the people who do the interpretation. And in that paper, there's something really interesting that is the use of these ratios of metabolites rather than the association to the pure metabolites. Can you say how you contributed to that paper, to that story, and then tell us a bit, especially about the ratios? I'm really interested in that.
Yeah, I'm pretty fond of that paper because it goes a little bit with, as I said, I moved to Munich and there was a professorship on bioinformatics and there wasn't really clear what I would be doing there other than teaching bioinformatics. And I went to the Helmholtz Center, at the time still called Gesellschaft für Strahlenforschung, GSF. And that's an interesting place because they host the Cora population study. There are different institutes. I was at the Institute of Bioinformatics, but there was an Institute of Epidemiology where Christian Gieger was working, and also his colleague Thomas Illig is also involved in that. And then there was a core facility where they were actually running, we had them measured by biocrates because we were setting up the platform, which was just I think it wasn't even officially on sales yet, the kit version, but we knew it was coming. So with Jurek Adamski, we were setting up the platform, and with Cora, we said, let's already generate some data, pay fee for service, measure 300 samples. And that's what we did. So we got a huge data set. I mean, huge, 300 samples at the time. It's not huge today.
At the time, it was huge, wasn't it?
It was, especially the kind of data was, I was very skeptical. For me, it was like, how can you measure a drop of blood and all having these details? How can that be precise? And then having another drop of blood and getting 500,000 gene variants out of that, and then do a correlation between them and find something meaningful. For me, it was a total surprise that
makes
worked.
You feel very small, doesn't it?
It's and there was certainly a lot of things in the data which was probably also not correct. I mean, you found a lot of things later on, misannotations of metabolites, which is totally normal. You have to know that. We ran the metabolomics against all other phenotypes of the Cora cohort, not only the genes. And we had I still remember we had a project meeting at some point where we had 15, 20 different phenotypes. And we actually split them up between all the postdocs and the guys interested. One would be working on smoking and one on alcohol consumption and one on diabetes. All the big topics that were in Cora, we split them up, and I computed the p-values and shared them with them. And I think there's a lot of papers that came out of that time. You can look them up, where we, for the first time, we saw associations between phenotype and metabotype. And many of them made sense, but many of them were also complicated to analyze. I mean, we had papers later on with coffee consumption, and we found a Bavarian nutrition style somewhere. Sometimes a bit long shots, but it was interesting to see that. And then the central thing, which was my project, was the GWAS with Christian together. The interesting thing for that one was, I mean, you need more compute resources. The compute center from Munich had the 10 fastest supercomputer in the world at the time. And they were very proud of that. But they needed users. And normally physicists went there and used all the time, but they needed non-physicist users. So at some point I got compute power from them, like, why don't you write a proposal? And I got compute power. And then they came, you have compute power, now use it. I said, how can I use that? How can I spend hundreds of thousands of compute hours on this machine? And that's where this idea with the ratios came up, that initially we did the GWAS already. And then we had so much compute time, and we had already done the idea with the ratios. We had it before when we looked at this original data we got from biocrates with the mice. I said, well, maybe we just try it out, because we just had the compute time. It needed to be burned. Yeah. And amazingly, it worked. I mean, generated tons of data.
So then with the computer there, you computed all the possible ratios between the metabolites. Yeah. Okay. Yeah. And then you checked which ones associated well with the data.
Yes. And then you look at this, what we call the p gain. So whether the p value really gets significantly stronger. Yeah. Because I mean, I can of course always do ratios, and if I do enough testing, I will always get a little bit of an increase. But what we found is, like, we had a p value of 10 to the minus 8 in the GWAS, and then it went to 10 to the minus 22. And then the very surprising thing was, like, the metabolites in the ratios. I mean, I tested all against all, and some people came like, why are you testing all against all? You should think before you do this. I really loved that, because the biology actually tells you what's right and what's wrong. It's not me coming up saying, I know that this and this metabolite are linked together. I just tested really everything, or also everything that didn't make sense. And the ones that made sense actually stuck out. So whenever I looked for something with a p gain, in the end, and also in subsequent paper, it really made sense. And in cases even further on, we had studies where we had unknown metabolites, where we didn't know what one of them was. We could actually, with a p gain, infer that this probably is linked to the other metabolite in the ratio.
Did you start building alternative pathways then based on this, with the few ratios that you did not expect? Did you look deeper into it to find? And you found the biology behind it?
Yeah, there was a paper with Jan Krumsiek on mining the unknowns from he was also one of the people involved at the time. And we could reconstruct pathways from partial correlations between metabolites and also from ratios, and reconstruct what biology was going on. So we had, I think one of our Gieger paper phrases was, if the function of FADS1, so one of our top hit genes, wasn't known, we could actually have inferred it from the data alone. And that's really something I found pretty pleasing, and something that very often replicated itself later on in larger studies. But it was there in the first study. And I think that's also why I like the first paper most, because it was just like you first describe things for a first time. Then we had another paper in Nature later. The first one we submitted it to Science and they didn't want it. So it just got to PLOS Genetics. The second time was easier, where I would have said, well, you already said that in the PLOS Genetics, but it went there, maybe also because it was bigger, of course, the study. And you brought again all the concepts there. And then it became a bit like, not all of the same, but of course it was more like generating more and more findings, which means there was not so much novelty anymore on the concept level, but the pharma, and not only pharma, but in terms of understanding biology, of course, there was contribution more and more. And even now saying, if one day we would have a GWAS on metabolomics in UK Biobank, conceptually, there wouldn't be probably not something new, but individually on the function of each gene, the overlap with the diseases, all that would all be the reason to do that.
And you think with in the case of this type of studies, like you have really powerful statistics behind it, like holding the story together. But do you find it makes it more difficult to publish when you have new approaches to, like a new problem and new approaches to that problem? It's not a good start, is it? Well, even though that's what everyone is trying to do, isn't it?
Yeah. Sometimes it's a bit too early for some ideas to get it really right away into papers like Nature and Science. PLOS Genetics was good, and I'm pretty happy about it. And the paper has almost as many citations as our following papers.
And the nice thing is that it's available to everyone as well. You don't need to have a license to read it. It's nice.
Yeah, okay, if you want to add that as well, yes.
Well, this is getting more and more, but Nature and Science are not famous for allowing people to let them read their papers. So for us, it's nice, for the audience. You mentioned that before computing all the ratios, you had already had the idea for the ratios. How did that come? Was it because you were interested in, for example, FADS1, and then you looked at the metabolites that were related to it, and then you saw a pattern there? Or how did you find out the first ratios, the ones that the computer didn't find?
Honestly, I don't even remember in detail, but it was on this mouse data that we that had from Biocrates, and it was Elizabeth Altmaier who was doing a PhD at the time with us. And I think it was like, how can we make sense out of this data? Just let's play with it in every way a bioinformatician comes up with. In the end, there was this first matrix where we looked at how the strength of the association starts, and we didn't really rationalize what it really is. Later on, we had a paper where we thought about what ratios really are, what they mean. But at the beginning, it was just like poking in the dark, if you want, and just say, oh, we could do ratios. Let's see what happens. And suddenly something came up, and then you say, oh, I can explain it. I think the rationalization came later to understand. I mean, to say a ratio could be a measure for the throughput of the reaction rate. That's one thing. Yeah. But then it also could be a normalizing factor. Like if you normalize with creatinine in urine, if there's like additional variation in your data set, like Fabian Theis from the systems biology group, he calls it the French fries factor. So if some people eat a lot of French fries and others eat little, then there's a certain lipid in your blood. And if you normalize by that, you reduce the variance. Yeah. And once you reduce the variance, the p-values get better. And that's the thing. So it's with a creatinine. You have a signal in urine, but if you have different dilution of the urine, you lose the signal.
Yeah, absolutely. Did you start out with a very defined workflow of how you wanted to analyze the data? For example, for the Gieger paper, like you had a series of things you wanted to try out, or did you have trial and error and then the kind of iteration process where you go, this could be improved with this, and this maybe we could do slightly differently because we saw the result is not really what we expected? Is this something that happens a lot in your work, or do you manage to go in a straight line because you know exactly what's going to happen?
Well, I would like to say it's the latter, but that's not true.
I don't think that's real, but I prefer to ask. You never know.
Yeah. No, I mean, the one thing which is really good with genetics is that you know that a genetic variant is causal, because the genetic variant cannot be confounded. In other studies, you can always be misled. You find an association. For instance, there are associations if you want to go for cancer. You do a case control study, you find something, and later on you find out that the metabolite you're looking in is a metabolite of orange juice, and you learn that the cancer patients get a lot of orange juice in the clinic. So then that's not really a marker for cancer. But in genetics, there's almost no way that something would confound the genetics. So if in genetic association studies, I tune parameters to improve the association, like how I normalize my data, how I scale my data. Of course, you have to be careful. There is always this risk every time you repeat a fitting and look at the p-value, you do another test. So if you're honest with yourself, you should not go for 0.05. You should actually have a list and make a tick mark. And every time you try it, you should make a tick mark and lower the p-value you should be hitting it. Realistic about that, but I think what I'm saying is, if especially an association is true, like for instance the one for FATS1, and later on you replicate that in another data set, and then you say, oh, should I log scale my data? Should I filter this way, that way? If I can tune the selection of my parameters to optimize an already known association without looking at everything else, and if I would then go and use that as an adjustment and then go and do the rest of the association, that would, in my view, be correct, because then I wouldn't look at the data. But the thing is, you have to be disciplined. You shouldn't tune your thing and say, let's tune all the parameters until I find the most hits possible or things like that. And that's, of course, the risk. You have to be honest with yourself.
Dangerous.
And that's a dangerous thing, because it's pretty frustrating to write a paper and say, I found something spectacular, and then it doesn't replicate. In the end, it comes back and bites you.
And this is something I also liked again in the Gieger paper. There was one part where I don't know if like this was the true chronology or if it was just written nicely, but there was this moment where it was like the association, the power of association was not good enough, and the p-values were too high basically for this kind of studies. So basically you go like, okay, so this would have ended here, but we found the ratios. It looks like, we would have been honest and we would have stopped, but we found the ratios and suddenly it was super powerful. But I liked this because it's a nice, whatever the story would have been otherwise, but like it's also a nice picture of a kind of limitation that you have sometimes when you do the analysis. And you find this sometimes in papers where people go, we tried this and it didn't work. But there are all these experiments or all those tests that are done that we never hear about, that are part of the work, that are part of the analysis, and that don't make it in the paper. Do you have an idea of the time you spend trying things that lead nowhere, as opposed to the time it took to do the actual thing that's ended up in the paper?
Yeah, I would make a difference here between a genetic association study and a clinical association study. For the genetics, the genetics is pretty straightforward. I think the way you do that is now pretty established. You can discuss how you scale your metabolites and things like that. You can maybe have more advanced things. You can do Bayesian, whatever association stuff, but that's more
quite routine
something now. And also I think in metabolomics you have so strong signals, you normally don't have to scrape the bottom of the borderline significant thing. So in metabolomics GWAS, we normally don't go for the borderline significant, although they could still carry information, but there's just no point in doing that, because the next GWAS with a larger sample size will catch these. However, for clinical studies, it's of course quite different, because you always start initially with the idea, I want to analyze my metabolome against this and this endpoint, and that you do, and that's okay. But then you start asking yourself, what is confounding? And you may find an association. Then you realize there's a weird metabolite coming up. And you said, this doesn't really make sense. I mean, we had a study where we did a diabetes case control study, and we took the controls from a dermatology department at the same time as the cases, which was a good idea, because we didn't have this kind of batch separation, taking samples at different places. But then in the end, there were some metabolites coming where I say, well, are these maybe rather marker for the controls? Because I mean, they were not really controls. They were like people at the derm department.
The ones who all got the same cream from the dermatologist or something like that.
Yeah, probably not, because it was very heterogeneous. But I mean, there was one marker that was actually melanoma associated something. Although later on, I think it replicated in other studies that were not like that. So maybe I was wrongly thinking it was wrong. But
but this
the
is
same
also
thing
the
is
thing
also,
with naming of, especially of genes. This can really be confusing if that's all you have to work from, because like genes are not studied much and there may be 1 or 2 papers that just led to its name, but not so much based on it. And then you think everything is brain. Do you ever have this?
everything is brain or cancer, because most of them are. No, and that's the problem with the genes. With the metabolites, it's a little bit less, although I think they're also, I mean, maybe now we jump from one topic to the other, but you come also in the interpretation of metabolite associations. I mean, this kind of pathway analysis, that's always something I'm always a bit reluctant as well, although that's also well liked. But it's also pretty tricky because you have so many pathways, and just saying, oh, this is a metabolite of oxidative stress. There is not one metabolite of oxidative stress. It could be indicative of oxidative stress, but it could also be a lot of other things. And I think that is what makes maybe metabolomics more hard to interpret than, for instance, proteomics. Because, I mean, proteins also have like multiple functions, but not so many. Some metabolites could be actually at the basis of almost everything. And to say this is a metabolite for this and this is a metabolite for that. Some metabolites, yes. But especially when you go to amino acids and like the more central thing, nucleotides, lipids, it's very hard to just pinpoint it to one thing. And then you also have the problem that the metabolites come from everywhere. So they could come out of the liver, they could come out of the kidney, from the fat system. And the thing is, and then again, there's maybe also a difference a little bit between other things you measure in blood, because the metabolites are in the blood for a purpose, most of them. So not all of them, but the organism puts them into the bloodstream to carry them elsewhere. But of course, you also have others that are there because a cell died, they were leaked or things like that.
Or they should be excreted later on. They're on
their way
out,
that's what I'm... Yes, exactly. You have the blood is a convoluted medium, and you have other medium, right? I mean, we have done things in saliva where we found interesting things like a marker for diabetes that you also find in blood. So you don't really know why does it appear there. The urine studies, people are doing CSF studies now. There's a lot of interest in this. But every time you have this problem of confounding with almost everything.
And with metabolomics, there's this extra layer also of what we eat, that's especially if you look in the blood, but also in any other metrics, like from when you start, there's, as you said, every organ contributes to the whole pool, but then there's also what we eat. And if the same person eats differently, then you might get different things, which makes it extremely interesting, but also very complex. And do you ever use microbiome data or diet-related data in your work with metabolomics?
Microbiome personally less, because we just don't have the studies for that. People starting doing that, especially in the twin study, they have several papers with microbiome already. So I think microbiome I could go on forever as well. I think there's a lot of catchy things there as well. The nutrition, yes, there's a study we did also with the same team in Munich years back. It's the HUMET study where we had 15 male healthy volunteers, and they went for 4 days closed into the Technical University Study Center, and they were 36 hours fasting, and they got controlled meals and everything. And Gabi Kastenmüller's group, they're just preparing a web server that now has all this data integrated. And there's also like the biocrates data, the metabolon data, the lipofit data from breast, from urine, from blood, of course. It informs you later on also about which metabolites are stable over the course of the day, where you wouldn't have to bother about fasting, and others where you probably would have to bother about fasting. Like this, there's a lot of confounding there as well. For instance, a diabetes person is more likely not to be fasting than a non-diabetic person, because a non-diabetic person could go without food. A diabetes person would probably be careful to equilibrate their nutrition intake, and then you would have already a confounding factor.
Yeah, absolutely. But this principle of making databases of metabolomes or other omes is a really interesting thing. I think you're working a lot on this as well, or to combine data about specific diseases or about specific topics to make it available to the community.
I didn't mention that in the introduction. We're working like in virtual group together with Gabi's group in Munich, and she's doing especially all this web server things. They have an atlas of Alzheimer's. They're just presently bringing up where they connect that. And the group of Jan in New York, they are very much into the systems biology of it. So the Gaussian graphical modeling and the networks, and how can we put p-values not only on a simple association, but on a pathway or on a part of the network.
Would you like to discuss some specific tools? There are tools especially to help make sense of the metabolomics or to integrate them with other omics?
I think in the end, it all comes down to people working on R and RStudio. And there are so many packages out there. I think new generation is also doing Python, which probably goes to the same thing. I'm not a Python person. I will stick with R. But I think both is pretty close to each other. There's certainly a good way of looking what other people publish, because there's so many new tools out there. A lot of stuff is how do you visualize your data and how do you nicely produce it? So going away from what I personally don't like too much is these hairy balls that come out like here's a network, there's a network, but you just see it and say, okay, what do I make out of that? I'm always thinking about my medical collaborator. Would he really pull something out of what I'm putting into that paper, or is it just to show that I have big data and I can manage it and nobody else can? I think there's a big gap there between what the bioinformaticians and the systems biologists can do and what the clinicians, who actually are our clients in many cases, really do with it. They come and commission a metabolomics study, or we do it together. They say, I want to do metabolomics on them. And then you run the whole thing and they have their data and you give it back to them, and then they're frustrated. They say, oh, yeah, so what next? What does PCAA 36.4 associates with smoking? What does that mean? Does it have any implication? And many people walk away pretty frustrated, to be honest, on that. And I think that's something we still have to really work on, to nail things down and to say what is really a takeaway message, more than just saying this metabolite goes with that and this metabolite goes with that.
This is exactly the purpose of this project and also of the podcast, to give people clues. So the clinician or the research scientist who is not necessarily an expert in metabolomics and wants to understand these results. So what are ways to help people to jump that gap?
Let me list a few things. It's more like throwing buzzwords out there. I think one key word is certainly Mendelian randomization, which is using GWAS data to build causal relationships, or infer or confirm causal relationships. So if you have a lot of GWAS data, big GWAS data with much power, then you could, in a way, show that a certain metabolite is on a causal pathway to a disease, which would then mean, in terms of causality, if I tinker with that metabolite, I would change the outcome. And that's what you want, right?
When you work with this kind of associations, like you build onto the kind of better annotation of the genome to try and understand where the metabolites fall, or?
Right. The thing is, I would have an association with a disease like diabetes and an association with a metabolite. And I would try to figure out, is the metabolite something that causes the disease? So if I bring down a certain value, would that improve the disease? Or is it a consequence? So it might be a good biomarker, but it doesn't make a sense to target that, to change that value. So that's, for instance, I think something that is big at the moment, and it really requires even larger GWASs on metabolomics. And I think that alone justifies the effort. Second, there's, of course, the ratios, which I still strongly support. Gaussian graphical models, GGMs, or so-called partial correlation networks. That is really interesting, because if you just do correlation networks, you end up with a hairy ball of everything connecting everything. So that's something I would look at. You can predict variation in metabolites in people based on their genome, only the genetic part, of course. So you could do that and link that to disease. What I haven't mentioned yet, we have in the meantime also done EWAS, so epigenome-wide association studies. And that's something which complements, in a way, GWAS, because GWAS is genetic. That's from birth, you're set to be a fast or slow metabolizer for this and this gene. The EWAS, as I see it, is like there are certain parts in the genome that get methylated, and that are switches to switch on or off genes. And in some cases, they reflect what your body does. So there's a very strong association of TXNIP with diabetes. And I think it's a read of how much of TXNIP the body actually needs to cope with the diabetes. So it's not necessarily creating the diabetes. It could be the inverse. It could be showing you what the body just does at the moment to cope with the disease. And that's a tricky thing. Is it a sign of disease or is it a sign of the body coping with the disease? But it can give you ideas on where you would go and try to treat the body. And then also a way of maybe early markers, because if your body is adjusting and fighting diabetes, but you're not diabetic yet, but you have already your transcription profile is switching. And we did an EWAS paper later on in 2016 with Ann-Christine Pettersen, where we did an EWAS on metabolomics with the same data, just like before. We found 20 different genes where the CpG was associated with metabolites. And independent of genetics, there were others where the genetics was confounding, but 20 that were that. And a large part of them, I think 8 or so, they were all associated with smoking. So there was already a marker, because you had marker of, I think smoking is a very good signal there. And then we had a few isolated ones. Initially, I didn't know what they had in common. And I think the message, and they made a message for people analyzing data, was we were always looking at the top hit. And there was an error, because it's not only the strongest association that informs you, it's all the metabolites, what we call the metabotype you should look at. And when you looked at it later, again, we did an EWAS with BMI and with diabetes in a Qatar cohort. And it turned out there were actually 3 of these genes. They all had the same metabotype. And like 15AG, a marker of hypoglycemia. And they had been reported in the paper before to be a metabotype of diabetes. So all these 3 genes were diabetes-associated metabotypes. And only later, people discovered that these genes were actually associated with diabetes and with obesity. So our EWAS actually had the information already. We just didn't see it.
No,
that means, but it's also, I mean, it's also a good thing, because it means there's a lot of information in your data set you could be looking for. And I think especially for people working on metabolomics, it should be very motivating, because there's a lot of stuff hidden in the data that is waiting to be digged out,
and that you can't really see yet, because you have been, especially if you don't work with associations, but you work just based on the previous knowledge of biology. Then there's a lot of connections you can't see, because you just can't right now. It hasn't been discovered yet.
There's another thing which I always like to do is, when you have your data, normally you collect covariates, age, gender, all that thing. I would always looking at associations that I know of and use them as as positive controls, so that you can be sure that your data is having the information. And for instance, if I have a previous study, and I know how strong the association with age is, and suddenly my association is weaker or stronger, I know there is something going on. And also to do association with things you expect not to associate. To do an association with a random number, it should always come out negative. But if you say, I do an association with the day of data collection or all these technical covariates, I mean, that's very important to not just throw them in as covariates, but to look at them and to make sense out of them.
Yeah, this is a very strong point for annotating the data with as much information as possible, and not removing the metadata that is given to us, because, it doesn't matter if it was a Tuesday or not, but maybe it does.
And I have interesting examples. So we did a multi-center study on kidney rejection, and there were metabolites that were center specific. They were only measured in one of the clinics. And when I looked at the thing, it was totally perfect, because the different clinics did the initiation of the immunosuppression with different drugs. So the metabolomics actually measured the drugs that the clinics used. I could have reverse engineered how the different clinics initiated the immunosuppression. And we had other studies where you would have unknowns that would just go like, I would order the samples in order of collection, and the metabolite would suddenly disappear or appear. And when you looked at that, you say, oh, we just changed the urine tube, or the labels on the blood tubes were changing. They were 6-digit numbers, and suddenly they were with the letters, which mean they had used another brand. It doesn't mean that your data is bad. Metabolomics is so sensitive. In the urine tube, it turns out they have some kind of preservative in there. And probably the other tube has a different preservative. And it probably doesn't make any difference. You just have to be sure that you don't use And you mustn't use the one tube on the cases and the other tube on the controls. But then
also,
then it doesn't.
As I mentioned before, I try to make a point for the place of creativity in interpretation of data. Do you see a place for creativity in your work? Do you think it's something important for a scientist in general and for the type of work that you do in particular, or what's your view on this?
I mean, if you mean with creativity, ideas, definitely. If it's for creating false data, definitely not. But
I don't mean faking data.
No, I think creativity, especially creativity in visualizing data, is important, because the most important thing is you really have to see things. If you only look at p-values, I mean, you may have very strong p-values, and once you look at it, it's just driven by 3 data points. That you can do with a simple plot. There's a new generation of informaticians who really take data presentation tools from totally different fields and bring them over into science, like from all this interactive Java visualization tools and things like that. And that's another thing. Things. Yeah, and the thing is also to be have your data interactive in a way, so that the user who isn't a bioinformatician could play around with the data in an easy way.
And it can also help to communicate your results if you have very dynamic study structures, like if you have different time points or if you have different stages of a disease, that can really help as well. Do you spend a lot of time playing around with visualization once you have the data, or at the beginning maybe, or at the end? I don't know.
Well, I personally don't find so much time for that anymore, but that's really where the PhD students and the postdocs come in. And I think where they also can make their mark by impressing their supervisors. But like, here is really something that speaks to you. You need to be convincing. So it's not like, it shouldn't be a black box, what you've done before. But in the end, the idea is, how do you synthesize a message out of your data? And there, of course, you can call that creativity if you want. It depends on how strict you are. Some statisticians would hate that, but
but I mean, as you said, in the visualization, you can be very creative, because for me, it is creativity to go from a black and white table with little stars to a graph that maybe has only 1 or 2 colors, but is putting the data in a light where you can actually see the differences, where you don't have to compute it in your head to see it.
You should create a hypothesis. It should teach you something, and then ideally you would either carry on with another experiment to prove that what your hypothesis generated is true, or at least, depending on what the study is, do replication, especially in mGWAS, to say, if I come up with something, I can be as creative as I want in the discovery, but whatever I come up with should replicate in an independent study.
I've had this in my own experience of analyzing data or looking at data sets, where sometimes, I did this analysis and then I did that analysis, and then I looked at through different angles, and sometimes I would forget to stop. And I consider this is a symptom of perfectionism, in a way, that you want to get to this kind of beautiful aha moment where it's like, oh, finally I've explained biology. But it's unlikely that you'll get to that ever. Especially with one study. Are you familiar with this? Can you maybe comment on this?
I think that's a very risky thing, especially if you don't have a fixed position, because it could mean that in the end you would never publish the thing and just get out of research, because in the end it's publish or perish in a way. I think it's not that, I don't see it that dire, but the thing is, you need to be there and tell a story to someone. You don't do it to file it away and when you retire, just say, I have everything in my drawer, but I never told anybody about that.
And do you think that statistics and significance alone can tell the story, or you need something more?
I think the most important is the statistics. A p-value always means what's the likelihood that you see the signal that you're seeing by chance. And if whatever you have in front of you could just be in front of you by chance, it's not worth it being considered further. So in the end, everything you do should, in one way or the other, be supported by statistics.
Yes.
That's something I like to do very often, is to just randomize the identifiers of my data, because that's what's also my biggest fear, that someone drops the box with the tubes and puts them back into the wrong order, and I don't know. That's why I said you have to test the age, the gender associations, to make sure that there's not something wrong there.
Yeah, that's a good point. I didn't know about this. Of course, I'm used to looking for outliers in groups and things like this. And we've had cases like this when I was working in academia where we didn't have so many replicates, but the signatures were clear enough that no, this is A and this is B, not the other way around. And they were like, panic, because at least we had clear signatures. But when you don't have this, it can be really risky.
Yeah. And that's important to have these kind of markers of especially sample integrity, because it's so quickly happening that, and especially if you work as a bioinformatician, never seen the tubes. I make a case of seeing them, but initially you just tell someone, they ship it, you don't even see them. And we had a case not on metabolomics, but on transcriptomics, where they gave a box of samples and they turned them by 90 degrees and took the tubes in that direction. And it was genetic, so it was easily spotted. And we didn't know what happened. And then if numerically we turned the thing back by 90 degrees, suddenly everything mixed. We checked the sex match at that point. So it was totally non-match. We turned them back and they matched. And I think this kind of thing, you like to have that for metabolomics as well. Of course, you would like to have somewhere replicates. Although if you go for this fee-for-service thing, it's hard to just send 3 times the same tube. Sometimes initially we did that. We even sneaked in a few samples and didn't tell the people there were duplicates in there. It's not really that conclusive, because the thing is, especially if you sneak them in, it's hard to bring it up later. That's the thing. You want to have duplicates. You want to have measures for your CV, the variants, technical things, everything like that. And I think that's also, especially when you work with core facilities and fee-for-service providers, you trust them to a certain point. And once you create trust with them, you prefer working with their data rather than with others, because suddenly there comes this other paper and say, oh, we used mass spec metabolomics to measure this and this, but it comes from a platform I've never heard about, and I don't know what to think about that. I would like to see all this kind of standard things done before, and would they replicate the FATS association? Do they see the same?
It's similar to comparing methods for other types of scientific research as well. For the omics, we always kind of expect any omic to be comparable to each other. And sometimes it's like this, even though, of course, you have points where you can compare and you have the quality controls, that it's probably more robust than other kinds of biological research, but still. There's, and
that's also why we should not be too shy of replicating things. Everybody wants to be the first to do that, and the second paper, the editor says it's not interesting. But I find most often the second paper is the one that really confirms it's safe to say, and you discuss what is really replicated and not what the first group just stretched to the end of how far you could go in your interpretation. Also, with more data coming out, I mean, the community is growing. It's a good time now to start in the field. And of course, also in getting more standardization, there's also a gap in the community there. Not a gap, but I mean, the people who are really the mass spec specialists, and then you have the people who analyze the data. And between them, there's sometimes the mass spec specialists are not so much concerned about what people do with their data later. They want to be as precise as possible. And then you have this kind of, the best could be the enemy of the good, because having a 10% coefficient of variance is not bad for something, but for someone really biochemist, it might be already horrendous.
Yeah, it's all very relative.
Yeah.
Is there anything you would like to add on the topic? Maybe a message you want to get out to the public? No, nothing serious.
I could end with my favorite quote that I always put under my emails. That is, if you torture the data long enough, it would confess to anything. And I think that's really something I always put it there to remind myself. You can find everything if you just look at the data long enough. In the end, we always are responsible to the people who read our papers, who are not metabolomics people, who are critical to what we do. While we have to be creative in our methods, we have to be critical as well as what we claim out there.
It was delightful to talk with you. Thank you very much for your time.
And I wish you good luck with your book and everything.
Thank you for joining us in this discussion. I hope that this episode gave you new insights and ideas on how to plan, conduct, and communicate your own metabolomics projects. If you'd like to continue this journey with us, make sure to register for the Metabolomist email list on the podcast webpage. For regular news on metabolomics and data interpretation, you can connect with me, Alice Limonciel, on LinkedIn, where I post on metabolites, analysis strategies, data processing tools, and more. And make sure to check out our other podcast episodes on the Metabolomist website.